Today I would like to represent one of our latest work entitled the NF-kappa-B regulated kinase SNARK mediates genomic instability and mitotic dysregulation in malignant melanoma. Outline, the SNARK, which is a stress-inducible kinase under the transcriptional control of NF-kappa-B, is an emerging oncogene. NF-kappa-B SNARK acts as a melanoma. The centrosomes and the relation to the chromosomal instability and cancer, and the impact of targeting NOAC2 on cancer cell cycle. The presence of abnormalities in structure and number of centrosomes has been frequently correlated with cancer. Abnormal spindle assembly and inappropriate chromosome distribution lead to genomic instability, which are hallmarks of cancer. So cells must precisely coordinate their centrosome cycle along with the cell cycle. NOAC2 has been already published to be upregulated in malignant melanoma, but no one searched for the molecular mechanism. Here we aim at unraveling the molecular role of NOAC2 in the melanoma and other types of cancer. NOAC2 kinase, or the SNARK, it is an AMP-dependent kinase-like kinase. It belongs to a family, big family of AMP-dependent kinases, about 13, regulated by the master kinase LKB1 and located on chromosome 1. It is the only kinase in microarray studies that found to be upregulated in response to stimulation with tumor necrosis factor alpha. Also, it has anti-apoptotic activity under the transcriptional regulation of NF-kappa-B. Its substrate, the MEP1, has been involved in a raw GT-based pathway. That signaling pathway has been always linked to cancer, and especially metastasis and spread of cancer. So we asked how NOAC2 can control melanoma proliferation, maybe by abnormal mitosis that finally or ultimately will end in aneuploidy and tumorigenesis. Here we checked for the subcellular localization of the emerging oncogene NOAC2 at melanoma cells. We found it is located at the centrosome, and we co-stayed with the marker of the centrosome, the pericentrine. Centrosomes in melanoma have abnormalities already have been published, so we found multiple centrosomes, abnormal centrosomes, non-separated, fragmented. Both have expression of NOAC2 at various phases of the cell cycle, interphase, prometaphase, and also going to metaphase, anaphase, and finally cytokinases. As you see, abnormal pericentrine and abnormal NOAC2 in melanoma cells. We furtherly also confirmed, previously we have confirmed that the NOAC2 is an integral part of the centrosome located as a centrosome in a cell cycle dependent manner in MEF cells, HeLa cells, and other types of cancer like colon cancer. By Western blotting, we could detect the protein expression of NOAC2 at various stages of the cell cycle, as we see from the FACS profile of the cell cycle. But the latest expression is at G0 of the cell cycle, and after the completion of mitosis, little bit decreased expression, but still present at the cell. We confirm it by Western blotting and centrosomal separation that it is an integral structural part of the centrosome. Overexpression phenotype yields multiple centrosomes, makes phenotype of polynuclear cells, polyploidy, and the cytokinases defects that also in turn will lead to multiple centrosomes. Interestingly, the overexpression of the kinase mutants leads to disruption, many mitotic defects, spindle abnormalities, fragmented centrosomes, as we can see here, fragmented pericentriolar material. The pericentrine is fragmented. So we can conclude that the kinase domain is needed for the function of our kinase, particularly during mitosis. Here we gather and we notice the many abnormal centrosomes in cells expressing the kinase dead mutant of NOAC2, ranging from structured centrosomes, multiple centrosomes, here multiple centrosomes, here structured centrosomes. It's variable. And we wondered what will happen to the cell cycle of the tumor cells if we knock down by small interfering RNA NOAC2. Interestingly, the cells stay at metaphase as we confirm it by immunostaining with the pre-anaphase marker cyclin B1, phosphoserine 10, phosphohistone H3. So this is the cell cycle profile of cells lacking NOAC2. They arrest at the G2 boundary and not go through the normal cell cycle. Also, the protein was successfully depleted by the interfering RNA. We found apoptotic DNA changes after knocking down NOAC2, ranging from multiple nuclei, small nuclei, nuclear fragmentation, dumbbell and knob-like structures. So depletion of NOAC2 in various tumor cells will ultimately end with apoptotic changes and the cells arrested at metaphase, failure to complete their mitosis. Then we wonder what is the effect on the centrosome. Is that NOAC2 is also essential for the normal cell cycle away from the cancer cell cycle? Yes, it is needed for the cell cycle of the centrosome and normal centrosomal duplication that happened during the S phase of the cell cycle. So if we look here, defective separation compared to normal separation of the control experiments, and mainly cells lacking NOAC2 will remain having a single centrosome, maybe filled to double. During S phase of the cell cycle, as we synchronized by hydroxyurea treatment, also we noted fragmented centrosomes. So targeting NOAC2 can lead to many phenotypes affecting the centrosome itself, ranging from fragmented centrosomes to unseparated centrosomes, and of course will be followed by abnormal spindle formation. So we can sum up that NOAC2 is essential for the centrosomal integrity, and targeting NOAC2 by small interfering RNA can cause centrosomal fragmentation, and also NOAC2 is essential for centrosomal duplication during the S phase in normal cells. We search for the interactors of NOAC2 and the centrosome and what makes NOAC2 control the aggressiveness of cancer, and we found it. Interactors, the famous oncogene, has been noted in many types of cancers, including melanoma. NOAC2 interacts with PLK1, the bolo-like kinase, the famous oncogene, has been also proven to be associated with aggressiveness of melanoma, and many anti-therapeutics targeting NOAC2 has been in clinical trials in treating melanoma. So by IP Western studies, they associate with each other. So NOAC2 associates with PLK1, so it might affect this oncogene and mediates the control of the mitotic cell cycle through this link. And if kappa B stimulates the NOAC2, then NOAC2 interacts with PLK1. This PLK1 has two major important sites, phosphorylation sites, the serine 137, and we wondered what will happen to the phosphorylation sites if we knock down NOAC2, and interestingly, that phosphorylation sites was decreased, but mainly at metaphase cells, not at telophase cells compared to the control experiment. So we scored the targeting NOAC2 or knocking out NOAC2, knocking down NOAC2 will be associated with decreased phosphorylation of PLK1 at the serine 137, mainly at metaphase centrosomes. increase the phosphorylation of another site, threonine 210. Also the same, we found it is decreased. And on the other hand, the increased immunoreactivity of MEP1, the famous substrate of NOAC2, that actually inhibited. It is a phosphatase that dephosphorylates BLK1, and it is inhibited by NOAC2 in its presence to allow for BLK1 to proceed for mitosis. Now we are knocking down NOAC2, MEP1 is overactive and dephosphorylates BLK1, and the cells will be arrested and cannot continue mitosis. Also, MEP1 is increased expression, and if we count the pixel intensity, this is the NOAC2 suppressed cells, and they have higher immunostaining and immunoreactivity of MEP1, the substrate, as interphase, metaphase, and as a kinetochore that is essential for normal segregation of the chromosomes. So our hypothesis, what we reach after that experiment, that NOAC2, the emerging melanoma oncogene, if we target it by drugs or small interfering RNA, releases the inhibitory phosphorylation of its substrate MEP1. MEP1 in turn dephosphorylates PLK1, prevents mitotic progression, and leads to metaphase arrest. In the clinic, just to show up that the NOAC2 is also expressed in human samples, along with the B65. There is new NF-kappa B activation in samples from malignant melanoma compared to less expression in benign nevi, associated with higher elevated levels of NF-kappa B circulating levels. And we conclude that in NOAC2, in turn, due to increased ROS stimulation by NF-kappa B, is highly expressed and drives the progressiveness in melanoma cells and maybe other types of melanoma. Here is mainly the pro-GTPase pathway and NF-kappa B pathway stimulate NOAC2 that in turn inhibit MEP1 and affect the inhibition on BLK1, so mitosis goes on. Our model is the NOAC2 overexpression, especially in cancer cells, will lead to a variety of abnormal centrosomes, apparent cytokinases, tetraploidy, finally aneuploid progeny, and oncogenesis. Thank you.